Hi friends, so today let us learn about antigen antibody interactions. Whenever there is an antigen and an antibody, there will be a serological reaction. So we will be first dealing with what is affinity, what is avidity. Affinity means that there is a intensity of attraction between antigen and antibody molecule is called as affinity. Avidity means it is the strength of bond between this antigen and antibody in an antigen antibody complex. So in an antigen antibody complex there will be a bond between this antigen and antibody, and how much strength, that is avidity. Okay, precipitation. First, antigen antibody interaction. Okay, precipitation, here we are adding a soluble antigen as mixing with an antibody to give insoluble precipitate. That is precipitation reaction, and the basic example you can remember is Ascoli's thermoprecipitation test, which we are carrying done for anthrax antigen. Okay, so that is precipitation. Now first we will deal with ring precipitation test. So what you are seeing here, that is a ring precipitation test. The solution contains antibody and above you are adding some antigen. Then what happens? At the junction of those two liquids there will be a ring formation where there is antigen antibody interaction. There is formation of precipitate. So in precipitation reaction, a soluble antigen is mixed with antibody to give an insoluble precipitate. So you can see that at the junction of the liquids a ring is visible. Okay, so what is the mechanism of this precipitation reaction? What is happening in this precipitation reaction? That is actually in the antigen and the antibody. So where you have the optimum concentration of both antigen and antibody, there the lattice it's complete. There will be formation of a lattice and there will be precipitation. So there is a zone of equivalence, and there is, so what happens when the antigen is excess or when the antibody is excess? There will be no precipitation. Precipitation occurs only when there is optimum concentration of antigen and antibody. If the antigen is excess or if the antibody is excess, there will not be any precipitation. So the zone where you have excess antigen is called as prozone. That is post zone, and the zone where you have excess antibody is called as prozone. So prozone is the zone where you have excess antibody, and post zone is the zone where you have excess antigen and where the precipitate is formed. That is zone of equivalence. There the lattice is formed. Now in precipitation reaction, so this is ring precipitation. Now you have something called flocculation. Okay, so this precipitation actually it can happen in any medium, in liquid medium or in solid medium. Now when it happens in liquid medium, that is flocculation. That means actually what is the difference between these two? In precipitation, after the formation of a precipitate, that will get sedimented. In flocculation, they will appear as floccules. Okay, that's a modification of precipitation. Here you have slide precipitation and also slide flocculation and tube flocculation. And the typical example for slide flocculation is VDRL, done for syphilis, and there is something called Kahn's test which we have done previously for syphilis. That is the example for tube flocculation test.